Well, thanks, Jonathan. This is a very personal introduction. So it's terrific to follow Alan's talk because Alan did such a great job of introducing the idea of how difficult vision is, how long people have been trying to replicate the incredible functionality of human vision with computers. Vision has turned out to be one of the most difficult information processing problems we've ever identified. So Alan talked about some of the reasons that vision is so difficult in the real world. Difficult because any given category of object is really infinitely variable in shape and pose. From time to time, you're going to have extremely different viewpoints on the same object. In the real world, there are major problems of partial occlusion of objects and clutter in general. Computers have a difficult time dealing with that. Human vision is remarkably robust to all of these problems, and that's why you all have to take these little tests called CAPTCHAs on websites, where the website is basically performing a little visual Turing test on you to distinguish you from a web crawling program to prove that you're a real human user, because in all of these cases, in spite of dramatic distortions or partial occlusion or clutter with other objects or noise in the image, it's always very easy for you to report the letters and numbers in the CAPTCHA. This kind of recognition can't be done in a simple way. If it could, then the summer project would have worked in the 70s that Alan talked about. It's not simply a matter of forming an internal template and then matching a retinal image to that. Biological vision must have a very intelligent, flexible way to solve all of these problems so effectively and quickly. And the project that I'll tell you about today, which is a new ongoing project, addresses what are the neural mechanisms, the learning mechanisms that make visual recognition so robust. This is a question on the algorithmic level, the same level that Alan was talking about. Rick really talked about the hardware level of memory. This is more the algorithmic level, that is how thousands or millions of tiny hardware changes at synapses add up to changes in the signals emitted by neurons that could be used for something like object recognition. So most of what we know about higher level vision at the algorithmic level comes from work on non-human primates, because that's where we can study individual neurons or individuated populations of neurons. At the same time, in an animal model that is extremely close to the human, has, for vision at least, extremely comparable visual capabilities and a strikingly similar organization of the many cortical areas that deal with vision. So this is a lateral view of the right hemisphere of a macaque monkey brain. And all of the little ellipses here with letters in them represent different stages in a pathway called the ventral pathway, that heads down into the temporal lobe. And it's this pathway of areas in the brain, in monkey brains and in human brains, that deals with object vision and to some extent scene vision. The black arrows between those different stages represent some of the major connections between stages. The ventral pathway, like all of cortical vision, starts out in the back of the brain in primary visual cortex, or V1, where cells process information about very small regions of the visual image, progresses through intermediate areas where cells process information about larger parts of the visual image, and eventually towards the anterior part of the temporal lobe, where there are cells that process large regions of the visual image, large enough to encompass entire objects. And I'll be talking about changes that occur with visual learning in this part of the cortex, that is the final stages of the ventral pathway. So this is the alphabet that we made up for this experiment. And like a standard alphabet, the identities of the letters are determined not by their exact shape, but what you could call their medial axis topology, that is their stick figure shape. And in fact, in the actual training and in the experiment, a given object, a given letter like this could be presented with very different lengths of the different elements, very different widths, and those are the kind of variations that we see in different fonts for the Roman alphabet, and we're very good at dealing with those. Also, like a standard alphabet, these characters are made up of simpler parts that are shared across different letters. This is a theme that Alan brought up, the idea of parts that in different combinations make different things. What you might notice is that all the rows here have the same bottom structure to the letters, and all the columns have the same top structure. So this is just a 4x4 matrix of top-bottom combinations. In this experiment, each monkey is just trained on half the shapes, and that allows us to set up the hypothesis that during learning, neural responses will change in a way that's adapted for discrimination between the trained shapes, and on the other hand, will not be adapted for discrimination between the unfamiliar shapes. So this is a diagram of the task that we train monkeys on. So this is the back of the monkey's head. He's looking at a computer screen. At the beginning of a trial, a dot comes on at the center of the screen. The monkey has to fix his gaze on that dot. Then one of these letter stimuli come up. There's a delay, and another letter stimulus comes up. And if that second letter stimulus is a match, then the monkey has to shift his gaze up towards a target that's always at the top of the screen in order to receive a juice reward. And if, on the other hand, the second letter is a non-match, then he holds fixation at the center, and that's how he gets a juice reward. Over here is a plot of the course of learning over days of training, 10, 20, 30 days, where the proportion correct responses for 4 different categories. The categories were actually introduced one after another. Is plotted. On day 1, behavior on a given category starts out at 0.5 or 50%. That's chance performance in this task. And it climbs slowly, and it takes about 4 weeks to get to a criterion of 85% correct. So this isn't an automatic thing. It's something that has to be learned laboriously in the way that at one time we all laboriously learned the Roman alphabet. So after training, we tested responses of individual neurons in TE, TE being one of the names for this anterior part of the temporal lobe. And the first thing we did was to test responses with learned shapes. So here are the 8 shapes that this monkey was trained on. And the y-axis here is spikes per second. That's the number of action potentials. This example neuron fired per second when each of these stimuli were shown. what we were looking for was neurons that responded in a significant way to at least one of these stimuli. That is significantly above background firing. Typical neurons will occasionally emit a random spike. For this neuron, the background rate was about 2 spikes per second. That's represented by this red dashed line. And there are a couple of stimuli that elicited a response that was significantly higher than that, especially this cross-7 stimulus, which elicited about 40 spikes per second. The area we were recording from was really an area that's buried in this sulcus here. This is an exploded view of the sulcus, so we're recording on the ventral bank right here. I plotted the ventral bank here. So this is lateral to medial, and this is posterior to anterior. The dots represent all of our recording locations. The blue dots represent locations where we didn't find any neurons that responded significantly to any of these shapes. And the green dots represent places where we found at least one neuron. Overall, 8% of the neurons that we encountered had a significant response to at least one shape. Now, these 8 responses don't really tell us much. We don't want to just know that there are signals for learned shapes in the brain. We want to understand how those signals are built up, how this neuron, through the network of inputs that it receives, is taking visual information from the retinal image and turning that into a reliable signal for this shape. That's always been a difficult kind of question. Because if you think about the entirety of possible shapes, the sort of space in which all shapes exist, it's virtually infinite. It's extremely high dimensional. We don't know what the dimensionality is. This is represented by this diagram here, where these arrows represent the dimensions of shape space. We don't know what dimensions the brain uses to describe shapes. You're trying to understand the information signaled by a neuron that responds in some region of that space. Over here, the space is so large that if you just sample it systematically or randomly, and that's what these red dots represent, random samples in this space, at best you're going to get one or a few samples in this region, and that's not going to be enough to tell you what kind of visual information the neuron is signaling or what dimensions of shape it's tuned in. So a way that we have been dealing with that problem is to start out with random sampling in this way, but then to create a second generation of shapes by partially morphing higher response shapes from the first generation. That represents small jumps in shape space, some of which are going to be off into the null response region, but some are going to be into regions where the cell gives higher responses. And then we iterate this process across many generations with the intent of getting dense sampling of this entire region, the peaks, the shoulders, the boundaries, enough samples that we can then fit quantitative models that explain to us exactly what kind of visual information these cells are signaling. So this is an example of how that kind of test works. So this is an earlier study. All these shapes represent the first generation of shapes used to study a single neuron in TE. We always divide this adaptive genetic experiment into at least 2 separate lineages so that we can confirm that they evolve in the same direction and converge on the same answer about shape. Here, the average response of the neuron to each of these shapes is represented by the background color, where black means 0 spikes per second or no response, and bright red would correspond to 70 spikes per second, which would be a very strong signal for IT. So here's first generation lineage 1, first generation lineage 2, and then here are partial family trees in lineages 1 and 2 that show how this adaptive evolutionary method works. So here's the highest response stimulus from generation 1. In generation 2, it gave rise to these stimuli, including higher response stimuli, lower response stimuli. Same thing happens here. Across generations, you wind up sampling the entire response range of the cell from around 70 spikes per second through 50, 40, 30, 20, down to 0. After 10 generations, about 1,000 stimuli, here are the highest response stimuli in lineage 1, and here are the highest response stimuli in lineage 2. And what we do is we take all of the responses, all of the stimuli, not just the high response ones, but the low response ones. We analyze them geometrically in order to fit a mathematical model that explains what geometric information is being signaled by this particular cell. And I won't go into the details of this model, but I'll just say that for this cell, the best fitting model involved two components. One represented in magenta, one in cyan, and those are projected back onto these high response stimuli down here. And the magenta portion of the model captures the sharp concavities positioned near the top of the high response shapes, oriented towards the right, and the cyan component captures the sharp convexities, also positioned toward the upper right and pointing towards the right. We can fit a model to lineage 1 and do a pretty good job of predicting responses in lineage 2, and vice versa. So that's the method that we're now starting to apply in this learning experiment with the intent of finding out exactly what kind of changes in geometric sensitivity occur that allow for robust character recognition. So this is the same example neuron. This is the first generation of stimuli in one lineage for this example neuron. Here we're using two-dimensional shapes instead of three-dimensional shapes, since the learning stimuli are two-dimensional. Just as in the other experiment, higher response stimuli in earlier generations have a higher probability. Sorry, 1 minute left. Yeah, I'll need a little bit more time because you did tell me 30 minutes and it's really only been 19 minutes. Yeah, okay. Well, I'll rush. So, so this again is the process of second generation from first generation. After 10 generations, here are the highest response stimuli from lineage 1. Here are the highest response stimuli from lineage 2. Here are the lowest response stimuli from the two lineages for comparison. So first of all, it's striking that in this genetic algorithm that actually starts out from a random starting point, the guidance provided by the neural responses of the cell actually caused the evolution spontaneously of stimuli that look like the learned stimuli. But as is the case in all of our experiments, in the example that I showed you before, it's never the case that cells in the ventral pathway are simply selected for an entire object. The brain doesn't solve this problem by having a giant lookup table, and in fact, that would be mathematically impossible. Instead, the cells respond to configurations of partial shape that characterize many different high response shapes. So we again fit a model very quickly. In this case, the best fitting model has two excitatory components represented by these green icons, positioned toward the bottom of the shape. One, a concavity opening towards the upper right, one, a concavity opening towards the lower right. And the model also has a negative component that depresses predicted responses. Positioned near the top of the shape, a concavity opening towards the lower right. So the predicted response for this shape is high because it has concavities in the right position, the right orientation to drive the positive parts of this model. So predicted response 32 spikes per second, close to the observed response of 40 spikes per second. Similar result for this stimulus here because again it has matches to the two positive components. Here, in contrast, we have a stimulus that has shape fragments that match the positive components that should drive a high response, but it also has a shape fragment that matches the negative component. And that depresses the predicted response to 17 spikes per second, and the observed response was only 22 spikes per second. So this has led to what our working model of character learning is now, that neurons develop a kind of Boolean logic of shape fragments that allows for robust discrimination of characters. That is A and B and not C in this case. And that kind of logic based on shape fragments allows this neuron, for example, to do the difficult discrimination between this stimulus and this stimulus. These are highly confusable because they've got the same bottom structure. But the difference is that this stimulus has a sharp concavity near the top of the object, opening towards the lower right, and as a result, predicted and observed responses are much lower than the predicted and observed responses for this stimulus. So through that mechanism, this cell is actually able to give useful information that could be used to discriminate this shape from this shape and all the other shapes that the animal had learned. Notably, this mechanism wouldn't work on the unfamiliar shapes. It would give a high response to this shape that would make it confusable with this shape. And in fact, in the adaptive procedure, some of the highest response shapes turned out to look just like that. So again, this is an ongoing project. We're in the process of proving this kind of mechanism specific to the learned stimuli across many cells, across multiple animals. And future stages of this project will include using a new type of electrode that allows us to record reliably from the same neurons across weeks or months so that we can follow these tuning changes through time as learning occurs. And also using microstimulation to stimulate these neurons and try to bias perceptual decisions. So, and then just quickly, the people that have done the work, Satch Sokol started the project, coded up the stimuli, did the most of the recording from the first monkey and the initial analyses. Christiana Heisig is training a very stubborn second monkey. Andrew Chang is continuing recording and developing new analyses. I also showed an earlier study by Yukako Yamane and this work is supported by an NEI grant. Thanks.
We've got time for a few questions.
And thanks for the lovely talk. Is there a notion of grandmother cells for shapes? And is there an overrepresentation of beautiful shapes? You know, we all seem to, in art and beauty, we find things in common, and clearly in shape spaces, we love sculpture of certain kinds. Is there any notion of you seeing those overrepresented, whatever that means, whatever it means to be beautiful in shape space? Do you see evidence of overrepresentation in cells?
Thanks, Mike. So the first question, grandmother cells, which is a theme that Alan brought up, the idea that somewhere in the brain there'll be a cell that responds when you see your grandmother. So there actually are cells like that, but they're not visual cells, they're memory cells. So they're cells in hippocampus mainly, sometimes in cortex near hippocampus, that look like they'll reliably respond. This is based on recordings in epilepsy patients who have electrode arrays implanted prior to surgery to help guide surgery. Cells that will reliably respond to different views of the same celebrity, for example. But these are not cells that are operating in visual space anymore, they're operating in memory space. And they'll not only respond to pictures of the celebrity, they'll respond to the written out name, they'll respond to the spoken name. So I think in the visual system, because you need the flexibility to represent the virtual infinity of things that can be in the world, you can't afford to have a one-to-one like that. In memory, because there's going to be a finite number of things that you'll remember in your lifetime, a finite number of celebrities, for example, you can afford to devote individual cells or groups of cells to that. I think I'll skip the beauty part because that would be another talk entirely, but we have done work that you're familiar with showing that there is overrepresentation of three-dimensional structure that people find more beautiful, and it's another story. Sir.
Thanks for a very beautiful presentation. I'm trying to find out how do we correlate your research findings and extrapolate it to promote learning.
Sorry, could you repeat that?
I said, how do we correlate your research findings and extrapolate from it to promote learning amongst those who are visually inclined?
I think that's a great question. The relevance that I can imagine ultimately would be that learning to see is a matter of training neurons that put together part combinations like this, and you can imagine that training or retraining somebody who has difficulty with reading could be adapted for gradually building up sensitivity to these things rather than hitting people with complex shapes all at once, and by making training more particulate in that way, you could conceivably make it more generally accessible.
Should we thank Ed?